Hello. As I go through biotechnology in a couple of videos, this is going to focus on DNA fingerprinting and its applications. And then obviously the videos that are playing in the background, they might go into the topics in a bit more detail. This is an overview. Before one can do a DNA fingerprint, one has to have enough DNA. But given that the sample size might have been small, that could be a problem. And so a very useful technique is that of polymerase chain reaction, or PCR. Let's say that you get blood from a patient, or you find blood or a tissue sample at a crime scene or something, and you'd like enough DNA to test. What would you do? Well, obviously our cells replicate DNA all the time, and so you could try to mimic that so that the you have enough of a sample. And so therefore you're going to have to go through a couple of steps repeatedly. So the first step is your double-stranded molecule has to unwind. Now, in your own cells, you've got proteins that bind to an origin of replication and then make single-stranded DNA. But you can also then do that with heat. If you simply have a high temperature, close to 100 degrees, now you will separate the double-stranded DNA molecule into single strands, and then you'll be able to then make copies of the now vacant complementary strand. Now, you have about 6 feet of DNA in one cell. That's huge. And so if you're going to replicate DNA, you don't want to replicate all of it. That would just be an incredible amount. Once again, 6 feet of DNA in a microscopic nucleus. Very often you're looking for just a section of DNA that varies among the population, so you could perhaps identify an individual or the specific gene which is causing a disease. So you're looking to replicate that area. So a key then is to attach a primer which matches a specific sequence. So you don't want to replicate the entire human genome, just this little piece of it. Now, luckily, DNA polymerase can't just stick to single-stranded DNA and start adding new bases. It has to do so attached to a primer. So if you have a primer that matches the specific region of the chromosome that you are interested in, then you can only replicate that area. So this is a way of replicating specific areas by using a specific primer. If you're looking for whether your patient has a specific disease, you would use a primer that matches the region around the gene responsible for the disease. So you add the appropriate primer, and then now DNA polymerase can add new nucleotides. And so now you started off with one strand of and one double-stranded molecule, and now you have two double-stranded molecules. That's wonderful. Now, in the old days, to repeat this, you would then have to heat this to a high temperature again. And the disadvantage then would be this enzyme. Enzymes are proteins, and they require a specific three-dimensional shape. The high temperature needed to separate the strands would then potentially denature the enzyme, and then you'd have to add more enzyme at each step, which would just add to the cost and the labor which was involved. But then it was discovered that you could use enzymes from bacteria which live at very high temperatures. There are bacteria which thrive and in fact require high temperatures to live. They are thermophiles and hyperthermophiles. So one, such as Thermus aquaticus, lives at a very high temperature. So heating double-stranded molecules to a high temperature to separate it doesn't denature their enzymes. And so now you don't have to continually add new enzyme, which cuts down on labor, it cuts down on cost. So that made this much more practical. But now we have two double-stranded molecules that we then heat, separate them into their single-stranded forms. More primers bind just to the sequence of interest, and then more complementary nucleotides can be added. So now we have 4 double-stranded molecules with the sequence of interest. And then you can repeat this and make 8, and then 16, etc. Now, you can do this manually, and once or twice. I did this with classes where we just had water baths, and then the class, then had your sample, and you would hold it in, this degree for 90 seconds so that this step could occur, and then you'd move it to this water bath for 60 seconds, and then it would be here for 3 minutes, and then you go back, and so you can do that. So if steps A and B are occurring at different temperatures, you can manually change it in 3 different water baths. But then obviously far more efficient is to get thermal cyclers, which then do it for you. So what you can do, say you're leaving to go home from work at the end of the day, you can load your samples and then program the machine. We'd like you to do step A for this amount of time at this temperature, step B for this amount of time at this temperature, step C, same thing, and then repeat it this many times. And then when you come back the next day, you will have enough DNA to sample. All right, so polymerase chain reaction, this is a precursor to the DNA fingerprints because it gives you enough of the sample to test. The second step before we get into DNA fingerprinting is obviously if the DNA is present in one long molecule, it will move very slowly and it doesn't tell you much of anything. What you want is to look at, to cut the DNA into pieces and then to compare the pieces from various samples. The problem with that is there are lots of enzymes that break DNA into pieces. Your digestive system does it all the time. You break down the DNA of the things that you eat. But it does it at random, generates different pieces each time, and ultimately just breaks it into nucleotides and nitrogenous bases. So it's not useful and it's not repeatable. But bacteria can have enzymes which are not trying to digest DNA. They are trying to disrupt viral DNA. So the theory then is a virus might enter the bacterial cell and have a sequence of nucleotides that the bacteria doesn't have. So if there's an enzyme that only binds to that sequence and breaks it, that would inhibit the virus, but it wouldn't affect the cell. So these are called restriction endonucleases, and they bind to specific sequences. So they're very often named for the bacteria they come from. So EcoR1 would come from E. coli. And if there's multiple restriction endonucleases, so there might be 1 or 2 or 3 at the end. So this endonuclease will cut DNA, but only at a specific sequence, only where you have a guanine, adenine, adenine, thymine, thymine, cytosine, only there. And so therefore, this enzyme will recognize that sequence and make a cut. So this will help the bacterium defend from, say, a virus. But here's a way that we could now cut DNA in a predictable form so that only cut this sequence. And if you were to have multiple sequences which were identical, you would be assured that you would get the same cuts in each one. So you could study patterns because it's not the random process of digestion, but something much more controlled. And so if you had a DNA sequence which had one of the GAATTC, then this enzyme would only make one cut. So this top one only has one of those sequences. And so one cut would be made and only one cut. So this would make two separate pieces. If it was a linear piece of DNA, if we're talking about a circular plasmid, cutting it once would make one piece. Let's just say it's a linear piece. So this linear piece of DNA now has two, was cut at one site, and now we have two pieces when we do our DNA fingerprint. If this linear piece of DNA had two such pieces, then we would make two cuts, and that would generate three pieces. If we had more than that, once again, we're only cutting specific sequences. So the number of these restriction sites that we have would be the number of cuts, and we would generate that number of pieces. So now we have cut our DNA into a predictable number of pieces, and now we can do the DNA fingerprint. So the way you would do that is you would use algarose, which you would heat until it's liquefied, and then allow it to solidify. So just it's essentially the same as making Jello. So in Jello, you add a powder to water, you heat it up so it's liquefied. And then when you pour it, then it will harden. If you pour it into a mold, it will harden in the shape of the mold. And so you do that in a rectangular block so that now you have a gel made of the protein algarose in this shape to the width that you want. And you've added a little bit of comb, you've added a comb which adds these depressions. And so these depressions are now where you're going to load your sample. So you take a pipette and you add it to these depressions in your well. And this little piece of Jello-like algarose now has DNA samples in it, and it's in a liquid known as a buffer. The buffer is useful because otherwise things might vary. So the pH might vary, which could affect how your gel moves. As the DNA fingerprint progresses, you're going to lose some water, and that could change things. If there are enzymes which digest DNA, obviously, that's bad. And so you could bind the ions that those enzymes require. So in any case, so now we have this buffer, and we then put it in a gel box with two electrical leads. So there's a wire coming into one end and a wire coming into the other end. And then an electric current flows through the buffer and then through the gel. Now, you've loaded the samples near the negative end of where the negative lead comes in. And because DNA has a negative charge, the phosphates of the DNA molecules have a negative charge. Now the DNA molecules will migrate towards the positive pole, all right? You don't want to reverse it or put the gel in backwards, because now the sample would run the opposite way. So it wouldn't go through the gel, it would just go into the liquid and you'd lose your sample. So you want to load it near the negative pole. It runs towards the positive pole. And because you have pieces, the pieces will migrate. But it turns out that the big pieces will move more slowly and the small pieces will move more quickly. So let's imagine, that, here we have this one single piece that wasn't cut. Let's say it didn't have a restriction site. Well, it's pretty big, and notice that it's not moving very far. If there was one restriction site and we cut it to make two pieces, well, it would move farther. So we have two smaller pieces now, and they move faster. So notice that they're moving faster through the gel, etc. And so the smaller the piece, the farther it will move. Now, obviously, you want to stop this at a certain time, or else the pieces will just keep going once again into the buffer, and you lose your sample. So you check on this from time to time, and you stop it at the appropriate point. You then have to stain the DNA and then destain it. So very often you add a stain that turns everything a color, but then you destain it, and so that the stain leaves the algarose but remains stained in the DNA bands. And then you can look at it. Maybe you use a light box. Now there's different ways of doing this. Some of the stains are beautiful, like ethidium bromide. But you can use a UV light, and the staining is brilliant, the banding. But ethidium bromide could be a carcinogen. So if you're using a stain that binds to DNA, I mean, if you think about it, wait a minute, things that stick to DNA, I'm not sure I want to ingest that into my body. And so, for example, in my classes, we don't, we don't use ethidium bromide. It's beautiful, the staining, but, that would just add, extra precautions that we would have to take, to not, to be safe with the students. So then there are other less toxic things which bind DNA. So now we have this DNA fingerprint. What can we use a DNA fingerprint for? Well, a lot. So let me just go through a number of ideas. So first off, you could diagnose disease. So one of the problems is, let's imagine you're a doctor and your patient comes in and says, my head hurts or my stomach hurts. That's horrible, all right? Because what causes, a headache or something like only about 1 million things, all right? And so at a certain point, if you're going to try to diagnose anything, you have to test for things and maybe then start to rule things out, all right? Now, some of the things that you might test for are genetic diseases or predispositions to disease, especially if something runs in a family. If a family has a disease which, shows up, if cancer is more common in this family, if a close relative died of heart disease or had a heart attack very early, these are things that you might want to test for. And when we look at DNA and then later proteins, there, obviously mutations can introduce changes, but some of these mutations might affect the size of that section of DNA. So, there could be a variation in an intron, there could be an addition or a deletion, so that section of DNA, which is the variant, which is not normal, could actually be a different length. If the restriction site where you make the cut, if that gets altered, then in a normal allele, you make one cut, and maybe in the mutant allele, you make no cuts. So when you run a gel, there could then be a difference between the normal allele, which say has this size and runs this length, versus the mutant allele, which in this case is smaller and runs a different, and would then run faster. You could distinguish between someone who has normal alleles or mutant alleles, or someone who's a heterozygous and is one of each. Say your mother gives you a normal allele and your father gives you the mutant allele, then you would have each. So this individual would have gotten two normal alleles, one from each parent. This individual, 2 mutant alleles, one from each parent, and this person now got one of each. I then have, I give my students some, samples like how would you then interpret these with, diagnosing diseases. And obviously because, disease can be serious, things, I'm trying to be silly, here. So obviously the big green head is not, obviously a real disease. What would one do? Well, I ask my students to interpret some of these, and then so sometimes, some of this might not, so you might say, oh, I would interpret it this way or that way. But here, for example, if you are observing that this parent had, this DNA fingerprint and has this disease, you might then be interested in testing the rest of the family. Now, why would you do this, and who would pay for this? Well, I'm going to answer that, and perhaps this is a little bit idealistic, but I don't think too much. First off, if you were to have a predisposition to a certain disease, maybe philosophically I would just want to know. All right. Now, sometimes there's nothing that you can do about it, but like if I was going to develop, a neurodegenerative disease in my 30s or 40s, I'm older than that, obviously, I might want to know. I mean, maybe it would affect how I looked out on life. It might affect, my decision to marry or have children. If I knew that, come the age of 40, I would have, this difficult syndrome. So there's that. But then also, I mean, when you think of things like a colonoscopy or a mammogram or blood tests for ApoE alleles for heart disease, etc., why are you doing that? Because if you catch a cancer late or heart disease late, now not only is there a great risk to the patient, I mean, they might die, but any intervention might be rather expensive. All right, and risky and costly. So heart operation to put in stents, or bypasses, etc. That could involve long stays at the hospital. Or if a cancer is spreading, the chemotherapy which would address this. So that's bad for the patient. It's bad for the health insurance company that's paying for this. It's going to be a long, drawn out process, which is expensive. But if you could diagnose something early, all right, so if you could get a polyp and remove it while it's a polyp and not an aggressive tumor, if you could remove a mole which is acting behavior, oddly, just by freezing it, it takes 2 seconds before it becomes a cancer. If you could have someone encourage them to change their diet when they're showing the predisposition to heart disease before arteries get blocked, not only is this better for the patient, it's also far, far cheaper. It is in everyone's interest to diagnose disease early, or predisposition to disease early. And so let's say that one parent has a disease. You would then want to test the family, all right, potentially, depending on the disease, because this could help, the children, have lifestyle changes, with diet or whatever, which would decrease the risk of the disease. Or you might say, now we're going to do mammographies or colonoscopies at an earlier age, or dermatology visits. Like normally we recommend that you do this by age 50 or age 40, but because you have this, predisposition, we're going to recommend you do it in your 20s or 30s. And the insurance company, the perfect world, would be happy to pay for it, because these preventative tests are cheap compared to what missing this would potentially cost. All right, so if you look at this, here is a band of DNA that this parent has and this parent has. So if one's normal and one's mutant, this piece of DNA, that's not really helping us distinguish between normal and mutant. This parent has normal alleles. This parent has, and let's say we've amplified, a specific region of DNA where we know a gene causing this disease is located. This apparently is the mutant allele. So we know this parent has this mutant allele. You would then want to test the family, and here we have two children with mutant alleles. They are the ones who would then require further testing. Now, I'm making up this scenario, obviously. This child doesn't seem to have either one of these normal alleles, so that child, is, potentially very concerned about developing this disease. Might this normal allele then compensate? So if this is a recessive disease, might this be a normal allele? This individual is a heterozygote, so they don't develop the disease, but they could pass it on to their offspring. I'm making this case up, so obviously you need more information. But my students, I ask them which children would benefit from further testing. It would be these two. All right, they have the same mutant allele as that parent had. And so in my playlist, I have a number of these cases. So I have them where they're unanswered, and you can come up with your, interpret. And then I have a few which are then explained. And so, for example, if then we have ask, why did this child get the disease? All right, because the parents, they have this and they don't have this disease. They have the same band. Well, this then must be the mutant allele, and it must be recessive, because apparently this other allele can give you the normal phenotype. So if you're a heterozygote who has a normal allele and a mutant allele, that you don't develop the disease. So here's a wild type allele that prevents disease, as is this. And so each of the parents is a heterozygote with a wild type allele and a mutant allele. This child, however, got both of the mutant alleles. And so if that's the scenario, then this child developed the disease. And then if you tested siblings, which of those would be at greatest risk? Well, if this can prevent the disease and this one, apparently, because the parents don't have it, then these two children have a mutant allele, but they also then have wild type alleles. But this child doesn't. So that would then be the child which would then be the one that you would fear might develop the disease. And so if there are preventative measures or diagnostic tests that you might want to do those early. So just once again, big picture here, I have some more cases which are explained, but this is a way of diagnosing disease. You wouldn't know who was going to develop the disease until later in life when they actually had cancer or had heart disease. But if you can test this early, then potentially you could save lives, you could prolong life, and then you could have treatments which are far less invasive and far cheaper than waiting for the disease to establish itself. Another application of DNA fingerprinting is then to look at paternity tests. Now, back in the day, this used to be very time consuming, and courts were tied up with paternity cases. Like, how do you determine this? Like, that was what a lot of legal cases were trying to decide. And now it's not really. Only very rarely do you need to take this to court, because if this is the child's DNA in part comes from the mother. So this band of DNA in the child obviously then came from the mother. Now, this band didn't, because when a woman makes ova, an ovum doesn't have all of her DNA, just half of it. So the ovum which gave rise to this child had this piece of DNA, but not this one, right? So we don't see every band. But what you're going to look for is, where is there a piece of DNA that the mother did not give? So the child got this piece of DNA, but not from the mother. It had to have come from the father. The child got this piece of DNA, but not from the mother. It had to have come from the father. So of males who might be suspected as being potential fathers, which of those could provide all of those bands? So some of those, so if a potential, a father might contribute to some, that's not enough. It has to be all of the ones that the mother could not give, right? And if a father has this band, well, then we don't know, whether the child got the band from the mother or the father. Let's say there was a band here. But here you would say male number 2 could not have given this band, or could not have given this band, but male number 1 could have. And so that you would say, if those are the 2 candidates, that male number 2 is the father. So this is obviously valuable, because, if, individuals are considering starting a family, this might be important information. If there were legal obligations as far as support for the child, obviously this could be weighed into this as well. So here, the child has a piece of DNA that the mother did not give. Male 1 could not have given that piece of DNA, but male 2 could have. So then, that's a way then of solving paternity cases. So once again, for my students, I have some cases where they get to interpret a DNA fingerprints, and say, in this scenario, if we have a child and we have a mother, which of these 3 males would potentially be the father? And so, if you or your class wanted to, do the same, then by all means, you could use this. You could even try to use this to establish maternity, in that, here's an example, a child comes home with a couple from the hospital, and there was a question, was there a mix-up with the children at, the hospital? So one could, try to establish maternity as well. Other uses of DNA fingerprinting is, this is obviously a sillier example, but in this, if you listen to the audio that I have here, the question is, if the first child of this family developed a werewolf trait because unbeknownst, to the family at the time, the father had been bitten by a werewolf. And if you can identify the um, the normal allele for not being a werewolf and a mutant allele for being a werewolf, then the question can, then arise. If the woman then becomes pregnant again, will the child have the normal trait or the werewolf trait? Now, this is silly. Obviously, a lot of individuals, they are then faced with a very serious problem where the first child that they had their family had a serious disease. Now, this then gets complex very quickly. And so in a classroom, this could lead to discussions where there are no, potentially correct answers. And this is something that I'll stress in the next thing again. This is a tool. And then something I say to my class, a hammer. What is the morality of a hammer? And the answer is, it isn't moral. A hammer, you could use it to put nails in a roof. That's good. You could use it to hit somebody in the head. That's bad. We as a society, then we make rules. You can use the hammer for these purposes. You shouldn't use the hammer for these purposes. So once again, the hammer, the tool, doesn't have ethics. It's how you use it. And we as a society, we vote based on our morals and principles. What are the correct uses? So what are the correct uses here, and how can they be used? So let's just give a couple of examples. If a child was born to a couple with Tay-Sachs disease, it's a horrible disease, and it will take the child's life early in childhood, and there's nothing that can be done about it. There is no cure, etc. And so you could certainly sympathize that if a couple had their first child with this horrible disorder, it's recessive, so then each of the parents would be a carrier in this case. If a woman was pregnant again, the question would come up, will we have to go through that again? Will we spend the next 5 years watching our child slowly die and have nothing that we could do about that? You could certainly appreciate that they would want not to go through that again. And so they could then potentially have a DNA test of the fetus. There are ways of getting fetal cells. Amniocentesis would involve a needle withdrawing some fluid from the amniotic sac, some amniotic fluid, which would have fetal cells. Something known as CVS, choriotic villi sampling, involves a vacuum tube removing some cells of the chorion, that's an extra embryonic membrane, and getting fetal cells that way. And then you could then get enough fetal DNA to test. And if this child then shows that they would then have Tay-Sachs disease, the parents have a decision, and they could terminate the pregnancy because they might say, this is something that we choose not to go through again, and we would like to perhaps then try again with another child, another pregnancy, which would not have Tay-Sachs disease. Now, once again, in my classes, I would then throw out different scenarios. So what then, let's say we repeat this, but instead of being Tay-Sachs disease, which is automatically will result in death, with no hope of cure, how about cystic fibrosis? Cystic fibrosis is also a terrible disease, and it often shortens individuals' lives. But more typically, an individual might make it through, say, high school, but could live longer than that. Now, obviously, as a parent, you wouldn't want to lose a child in high school. You wouldn't want to go through that, wondering, will my child make it, this long? But obviously, we live in a world where lots of children don't get to live through high school. So, I mean, that would be a longer life than many children get to live. And so, what decision, could the parents, then, take? Would they, decide to terminate the pregnancy based on that? By the time your child gets to high school, who knows, a cure might be available. A third scenario is, let's imagine that, I'm being silly and making this up, there is a gene which can determine musical ability. All right, well, would parents say, we want a child who's very good in music, so we're going to terminate a pregnancy based on, on music ability? Like a lot of students will, perhaps come up with an example where, it's involving hair color, like I would like a child with, this, hair color. So once again, my role in my class discussions, I'm certainly not saying, right or wrong. I go back to my example. This is a tool. How it uses, how the tool is used depends on how society feels about this. The society makes the rules. And then I ask my students, A, you're parts of society. You're here taking a college genetics class, so you're better informed about these matters than many to most of the general population. What do you feel? And my students, debate this. They have different thoughts on the uses of this. And then, we can continue this conversation when we talk about criminal justice applications. So let's say there's evidence found at a scene of a crime, all right? And then you want to know whose blood is this, whose semen is this, whose skin cells are this, etc. And there are a bunch of people who are suspects who have been taken into custody or, are under consideration. Now, before there was DNA evidence, you could use physical evidence like, the fibers found at the scene of the crime from the rug match, something found on the pant leg of one of the suspects, etc. Or you could deal with eyewitness accounts. Someone says, I thought I saw someone who looked like you at the scene of the crime. A problem is that, they aren't perfect. Eyewitness accounts, have often led to incorrect, innocent people being, convicted of crimes, guilty people set free, physical evidence, the same way. And so now you could then ask, all right, if this is the evidence, here we have suspect number 1, 2, 3, 4, etc. Does this banding pattern match anyone else? And so here clearly it matches suspect 5. Now, that doesn't show that suspect 5 is guilty of a crime. If suspect 5's blood is at the scene of a crime or semen is present at the scene of a crime, they just simply have to explain how they got there, that there may be a good reason for their cells or their blood being at the scene of the crime. It doesn't mean that they've committed a crime. If there is a case where a woman says that she was raped and a man says that it was consensual, this is obviously a very difficult consideration. This shows that the semen from the sample matches male 5, but that doesn't answer the question as to being consensual or not. And so, this is not necessarily showing guilt. And an important thing to focus on is it can show innocence potentially, in that if the prosecutor is saying suspect number 2, that is your blood, the results could come back that no, it isn't. All right, there have been individuals who have been convicted, sometimes sent to jail, sometimes put on death row. And later DNA analysis has gone back and sampled the evidence, and it was shown you got the wrong person, which is a problem for lots of reasons. An innocent person is serving time, maybe been sentenced to death for a crime they didn't commit. And the person who did commit the crime is still free, potentially committing more crimes. The first time that this was used ever in England, DNA fingerprinting, and the first time that it was used in the United States, was used to exonerate someone. So a lot of people, automatically jump on, this is, showing guilt. It doesn't have to. It could show innocence. All right, because as eyewitnesses account, they've gotten it wrong. All right, and so if you did not commit a crime, it is in your best interest to test the sample, because there could be someone, I thought I saw someone who looks like you leaving the scene of the crime. That eyewitness account could certainly be erroneous. So you can use it in to identify. Use it in criminal justice cases. And I have some silly examples here where who was it who was wearing the robot costume, or which of these townspeople was wearing the costume if you find DNA in the costume? Who does this this match. And so, I asked my, students, who would you identify? And so here, person C has DNA patterns which would match that. So I have a bunch of, once again, silly examples, but obviously, identifying criminality at a scene of a crime, is very serious. There are then some interesting, applications as well. So, for example, here's a crazy case. Let's say that we're not testing, a sample, et cetera. Let's say that a person says, I could not have committed the crime because I was not there. I was somewhere else at the time. I was never at the scene of the crime. One case I remember, they tested pollen from plants at the scene of the crime, and then pollen from the clothes of the individual, and showed, so they didn't match the suspect's DNA to evidence at the scene of the crime. They matched pollen from the scene of the crime to pollen found on the person. So that is, once again, does not prove that the person committed a crime, but it now shows that they were there. And so there's all of these applications to this. And there's far more examples. So I just want to quickly go through some other examples of this. You can now study organisms, and compare organisms. And so if you can get DNA from other organisms, so for example, Neanderthal DNA has been isolated, another fossil human, Denisovan DNA has been isolated. And so you can come up with studies. So I have Neanderthal DNA in my ancestry. Fascinating, and so, you can ask, does, now that we know what DNA of Neanderthals looks like, do you have any in your ancestry? It turns out I do. If we know what mammoth DNA looks like and mastodon DNA looks like, if we then look at DNA of elephants alive today, we could ask, which are they more closely related to, mastodons or mammoths? What is their closest relative? And the answer would be mammoths based on this. So, you can study animals, even extinct animals. If you're studying animals alive today, there's lots of reasons to want to do this. So, for example, let's say you have rules against poaching, that you could breed something in captivity, but you can't take it from the wild. And someone has a bunch of lion cubs, let's say, and they say, oh, these are all siblings because I've bred them, on my property. I didn't poach them from the wild. But if they're all this different, then no, they don't appear to be siblings. They do appear to be poached from the wild. So, so then there would be, criminal, fines, et cetera, associated with that. If, you're served, you go to a country and you eat meat, and you ask, what is this? Is this meat from an endangered species, or, is this chicken? All right, well, if someone says, oh, it's chicken, it's not an endangered species, well, you can test that. If you were looking at an endangered species in zoos, and you have a female, and you would like to have this female mate with a male which is not closely related to her, so that the children would, the offspring would then be the healthiest. If you're trying to, save this endangered species, you could test the, you could then test these different males, and then determine which is least related to her, which would ensure that, the offspring are most healthy. So elsewhere I go into this more, but just biologists who study life, lots of reasons to now study the genes of a species, the degrees of relationships. If you're going to see, which bird should we put in the same family, which in the same sub family, how did this group of organisms evolve? So lots of reasons to do DNA fingerprints of wildlife. I'm just running through a bunch of potential applications. You can map positions in DNA. So for example, you could say then do if you have a plasmid of DNA and you're trying to map it, perhaps because you're going to insert a human gene onto this bacterial plasmid and have the bacteria synthesize a molecule that the hospitals need. You could say, all right, well, so I've cut my plasmid with this one restriction enzyme. Oh, and it turns out that restriction enzyme, let's say EcoR1, makes two cuts, a big piece here in yellow, and then a little piece there in blue. And then now I add another enzyme, and now I ask, where does it make a cut in this plasmid? If I'm going to now use this plasmid for some technique later on, I can map the restriction sites in the plasmid. So that's something that my classes do, and I have some videos explaining that. Now, one of the things that would be very useful if you are trying to map the a plasmid and figure out how big the pieces are and where the restriction sites are, is if you had a standard. And so you can buy that. You can say, hey, company, you sell pieces of DNA of known size. I'd like to buy that. And then I put that in my my gel. So one of my lanes are standards. So I bought DNA with a specific sizes. So here are the standard sizes. And so I know now when I run my gel that the lane that has the standards, there will be a piece that's 50,000 base pairs, 20,000 base pairs, etc. Well, why would you care? Well, if I then measure, how far did that piece travel? So, oh, the 50,000 base pair piece, that didn't travel very far because of how big it is. That traveled, say, less than half a centimeter. Here's 1 centimeter. Here's 1.5, 2 centimeters. So if now I can determine how far each of the pieces traveled based on their size, I could now make a graph, all right? Because I could now say, all right, how far you traveled, that's your X point on the x-axis. How big you are, that's on the y-axis. We're using semi-log paper, so the y-axis is on a logarithmic scale. That's why the lines look odd, but that means we'll get a straight line instead of a curve. And once I do this, I use the standards, and now I come up with a straight line which unite these points, which determines the relationship between how big a piece of DNA is and how far it travels on my gel. Once I have the line, I can now go back to my unknown samples. Remember the standards I bought because I knew I wanted to know how big they are. But now I've got all of my sample and I don't know how big these pieces are. They're unknown, but I'd like to know. Well, now I can simply say, all right, well, let me measure all of those. All right, something that travels 2 centimeters, I don't know how big it is. But now that I have this line that depicts the relationship between size and distance traveled, something that travels 2 centimeters is now about 10,000 base pairs. All right, so that's one application, and these videos explain it further. Another application of DNA fingerprint involves what are called dideoxynucleotides. So ribose, the sugar in RNA, has OHs on the carbons here, which are at including that one. So we can number these carbons. So if you study DNA, it does. So that's the 1 carbon, 2, 3, 4, and then 5 is attached here. So we're going to look at this 2 carbon here. In ribose, there is an OH, and in deoxyribose, there isn't. That's why DNA has that letter D. Deoxyribose is missing this OH. But then there is also another form of this pentose sugar known as dideoxyribose. Not only is it missing an oxygen here on the number 2 carbon, but also on the number 3 carbon. Now, because the chemical bonds between the carbons utilize the 5' carbon and the 3' carbon. This is a problem if you're trying to build a DNA chain because you get to a certain point where you stop, all right, because you don't have the OH group that you would need for the chemical reaction. So when we have nucleotides, which we can add in a DNA chain, here in red, these are the DNA nucleotides, which are missing an oxygen on the 2' carbon. Ribose would have an oxygen on the 2' carbon. But then you could also have dideoxy nucleotides. I'm going to use them with white sugars. They are missing an oxygen. They just have an H and not an OH on the 3' carbon as well. Now, that means that if you were trying to build a new DNA chain, so you had single-stranded DNA and now you were adding new complementary nucleotides, if you add a new DNA nucleotide that has that OH on the 3' carbon, you can keep going. But once you get to one where you add a dideoxy nucleotide, it stops because you can't, there's no OH group here that another nucleotide could attach onto. And so that would stop the chain. Now, you may be wondering, where are we going with this? Well, it turns out, and I'm just going to say what this can be used for, and then if you wanted to really understand how, once again, this is a playlist where these videos go into it. If you then had a mix of nucleotides that you were adding to a single-stranded piece of DNA, if you had some of the guanine nucleotides, which were dideoxy guanine nucleotides, then you would stop the replication wherever there was a guanine. So you would have some replication attempts which would stop here, and they would stop because once you add this guanine nucleotide, no other ones could be added. Now, some of the guanine nucleotides are the regular DNA nucleotides, so they would let you keep on going, but then you would stop at the next guanine, etc. And so and I'm sorry, here you would be stopping at the cytosines, obviously, because guanine's the complement. And so now you're getting an idea where are there cytosines, and then on the complementary, where are the guanines in this sequence, all right? And so we now know where the guanines, and their complements to the cytosine, where they are located. Now, if we do the same then for the other nucleotides, then what we can end up doing is determine the sequence. You now know where each guanine is, where each adenine is, thymine, and cytosine. And so if you're studying DNA, at a certain point, you know that you determine the sequence, that you now know here's where all of the adenines, the guanines are. And so you can say the sequence of my gene is GGACCC, because if we determine where each of these are, once again, I explain this. So when we added the dideoxynucleotides, some might have stopped at the adenine that was, and when we run the gel, we say, oh, that's only one nucleotide big. There's a thymine dinucleotide that's 2 nucleotides. And so we now get, okay, so here's the biggest, so this corresponds to this sequence, the longest one. So there must be an adenine here, there must be a thymine here. And if you know where all of the 4 nucleotides are, you could then say, oh, using this, I can now come up with the sequence here. It's adenine, cytosine, guanine, thymine, etc. I now know the sequence of my molecule. So when we do DNA sequencing, we use these dideoxy nucleotides in order to. Finally, there are so many applications of DNA fingerprinting. I'd like to just end with 2 more. So let's say you run a DNA gel. And you say, all right, well, I've got all of these pieces, but somewhere here is the piece of DNA that I'm looking for, but I don't know which piece it is. You could do what's called southern hybridization, where you could now kind of draw water through this buffer chamber through your gel that has the DNA here into what's called a nitrocellulose or a nylon membrane, which will trap the DNA. So the DNA you want to draw upwards, but it will get trapped here. But now let's say you have paper towels. You want to weight over this, which, the buffer is getting drawn into the paper towels. So you now—
the DNA will travel with this.
So you then pull your sample into this nitrocellulose membrane. So the DNA is now here. But now you could treat it with a probe that would stick to the a sequence of DNA that you're looking for. So you're asking, oh, I found this gene in fruit flies. Do humans have a gene like this? And so you've got something that will stick to that sequence. And you're asking, here's a bunch of human DNA. Do we have a sequence like that? Now, some probes are fluorescent. Some will change color, some are radioactive. So somehow there's a way of recognizing this. And you can say, oh, look, I found the DNA. There is a piece of this on chromosome 5, where humans do have a copy of this. And, so you pull DNA into a membrane, and then you can then, study it there. I don't want to say anything more, but once again, here's a video and a playlist where obviously I go into it a little bit more detail. And then in addition to being able to doing a DNA gel, a DNA fingerprint, and then studying that through say, southern hybridization, you could then do an RNA gel. All right, so put RNA samples here. And then, that could tell you something, but then you could do what's called a northern hybridization, where you're trying to look for RNA. Now, as I explained in this video, that's a little bit different. So we know that all cells have the same genes in an individual, but which ones are they using? Well, that's what the RNA could tell you, because some genes are on and some are off. So that, fetal cells or blood cells or brain cells, they have certain genes, but which ones are they using? Well, that's what the RNA could show you. Why is this cancer cell, acting abnormally? Is it using a gene that it shouldn't? So we know it has the same genes potentially as the normal cell, but is it now using one that it shouldn't? Is it not using one that it should? Well, this could then, show you, oh, bone, cells are using, this gene or that gene, or they have different lengths in different cells because of the way they're splicing their introns. But so in any case, so you don't have to just do DNA fingerprints of DNA. You could do the equivalent with RNA and study that. That tells you a lot about levels of expression. And very often that's important, which genes are being expressed. Is a cancer expressing something to an abnormally high level? You could also then do protein gels, where you're looking at what proteins are present, or you could do a Western hybridization where you then allow for probes there. So as we look at all of the applications of biotechnology, this was just kind of an overview of the basics of DNA fingerprinting, and then the some of its applications.